All right. I hope everybody is doing well. A lot of you have sent me your primary assignment that is due today at 5. If you haven't, please remember to send it to me. So today I have a few pieces about autoimmunity that I want to talk about just because I couldn't skip them. And then I'm going to move into hypersensitivity after that. The slide packet that I made, or the slide document that I made, is also slides I'm going to use on Monday. So I'm not going to remake it. Unless something dramatic changes, I don't really think it will. I'm going to do this for today and for Monday. So we saw a lot of information about mechanisms of peripheral tolerance and breaking tolerance in thinking about autoimmunity. And so we thought a lot about many of these mechanisms that are related to immune regulation or failure of tolerance in the past 2 class periods. That can all be associated with leading to autoimmunity. You also saw a number of examples last time of ways that infection can be involved in breaking tolerance and maybe involved with autoimmunity. But there are a few other things I just wanted to mention, largely with regard to kind of some of the other factors, particularly genetic factors, that may be important with regard to autoimmunity. And so I just feel like these are a few points that one should be aware of if one is taking an immunology class with regard to the genetics of autoimmunity. So there have been lots and lots and lots of studies that have done genetic associations with different genes and different disease states. I can totally stand on one foot, lecture, and tie my shoe at the same time. This is sort of showing an overview looking at sort of genetic linkages with autoimmune diseases as well as other disease types, which again, we can be picky about whether we're calling them hypersensitivity or inflammatory disease or autoimmunity like we talked about last time, but whatever. And you can see that there are some genes that might be linked to only one of these types of disorders. So here you can see, there's something on chromosome 13 that's related to asthma, but not to the others. But you can also see that there are lots of places where we see a bunch of overlap, where it's sort of the same genes that can be involved in many of these different conditions. When you look in general at genetic associations with autoimmunity, one gene particularly stands out. So it's the gene that you always see in any autoimmunity study. And I will admit, sometimes I will see people do these giant autoimmunity studies and they'll just come up with this gene at the end. And I'll be like, thank you, Captain Obvious. A little bit about it. And that one gene is MHC. So the MHC locus on chromosome 6 is always sort of the strongest link to autoimmune diseases, in that with different autoimmune diseases, there are either MHC types that are sort of almost always found, or in MHC types that are sort of never found. This seems to be related to what self-antigens are getting presented or could be presented on that MHC type to lead to the autoimmune epitope being recognized. And so the most common link that we see with different autoimmune diseases is particular MHC types. So I mentioned B27 earlier in the semester. B27, for example, is a really strong MHC type in terms of having a protective effect on HIV infection and progression to AIDS. Sort of like if you have are infected with HIV, your best prognosis is probably going to happen if you have HLA-B27. But at the same time, you can see HLA-B27 is also strongly associated with this autoimmune disease, ankylosing spondylitis. It's actually associated with quite a few autoimmune diseases. At one point, a rheumatologist looked at me and said, "I'm pretty sure you're HLA-B27 positive. I can just tell by looking at you." And I'm like, "Yeah, that's not how this works." Like I said, the MHC is sort of by far the strongest association. There are other genes that are pretty strongly associated with different autoimmune diseases. And so you can see some of them here. And what you'll notice is that even if we look at different diseases, which are shown in this table, we see that many of the genes that are associated with those autoimmune diseases fall into the same kind of groups. So it's sort of like always members of those groups. Specifically, you can see they are things like cytokine receptors or cytokines. So you have perhaps the wrong amount of a cytokine receptor or the wrong response to cytokine, or too much cytokine or something like that. Things like innate immune receptors, things like co-stimulatory molecules, things like proteins involved in antigen presentation. C-type lectins are another type of innate immune receptor. And so you can see it's sort of immune regulation types of genes that we see over and over again being strong predictors of sort of the genetics of autoimmunity. There is one other interesting relationship with autoimmunity that I just want to point out. It is an area of immunology that I could go way far down a rabbit hole on. If it's something that you're interested in, I already know one person in the class is, because I've already been emailing with them about it. I have a whole bunch of podcasts and readings about this if it's something that you find interesting. But if we actually look at different autoimmune diseases, by and large, we find that most of them are predominantly found in women. And so there is a strong sex bias in autoimmune diseases. So the table on the left is showing us a bunch of different autoimmune diseases and the ratio of females with that autoimmune disease to males. And so you can see there are some that have a little bit of a bias towards males, but the majority of them are largely found in women, and in some cases, like really dramatically largely found in women, more so than men. This is the version of the same thing from your textbook. What they're trying to show here is sort of how much more likely women or men are to have that autoimmune disease. If it's in pink, the women are more likely to have it. If it's in blue, the men are more likely to have it. And you can see how many of them have this bias in females, and in fact, how strong that bias is in a bunch of cases. This has been observed for a really long time. And there are a bunch of kind of hypotheses about what's up with this. And probably many of them are contributors. It's probably not just one thing. But just a few things I'll point out. Again, understanding sex differences in immunity is actually, like I said, a whole fascinating topic that one could go on a whole long discussion about. But one thing that we do notice, and this was sort of actually really the first thing that people talked about here, is that oftentimes we see autoimmune diseases in women after their first pregnancy. And so at some point after their first pregnancy, those autoimmune diseases get diagnosed. And so one of the things that we know is that the immune system changes during pregnancy because the fetus is foreign, and we need to not reject the fetus the way we would reject a kidney. And so the immune system has to undergo some changes during pregnancy. And so one idea is that your immune system changes during pregnancy to be able to maintain the fetus, and some downstream result of those changes may involve may eventually lead to autoimmunity. So that was kind of one idea that has been out there for a long time. And there likely is some truth to it. We also know that there are general differences in immune systems between men and women. And so this is showing immunity to viruses because, hi, look who you're talking to. I like viruses, but you can see this sort of throughout a lot of aspects of immune responses. And so in general, again, if we just look at immune responses to any antigen, any viral antigen, we see this pattern that is depicted on the left, where we often see that women have a much higher immune response than men do. And so in general, the immune response that we see in women is a bit on the higher side. The immune response we see in men is on the lower side. And as a result, it seems that in many different situations, women tend to be at higher risk of immunopathology, of sort of too much immune system, potentially autoimmune disease. So again, remember, immunologists love thinking about Goldilocks. And so women are going to be more likely to end up with too much because their sort of set point immune level is higher, while men are going to be more likely to maybe have too little immune response. And so they're less likely to have immunopathology, to have the immune system leading to damage, but they'd be maybe more likely to have pathogens persisting and pathogens kind of going out of control and leading to more damage. And so the damage might be coming from different places in these different individuals. And you can see that the table on the left is showing you a bunch of different viruses, kind of whether things are worse in males or worse in females. It's also kind of telling us, are we talking about disease? Are we talking about prevalence? Things like that. And part of this is, in some of these cases, the disease is really due to the immune system causing damage. It's due to the immune pathology. And so we're more likely to see that in women. In some cases, the damage is really due to the virus. And so we might be more likely to see that in men because they're going to have more viral replication. And so there does seem to be this difference that is sort of being understood more and more recently. There are two other pieces that we think are related to this. They're sort of part of what's going on with this. One thing that we've realized is that there are a lot of really important immune genes encoded on the X chromosome. There are a bunch of PRRs encoded on the X chromosome. We've talked about the common gamma chain, FOXP3, there's a ton of really important immune genes on the X chromosome. And in a lot of cases, that doesn't matter because in a lot of cases, cells in people with two X chromosomes will undergo something called chromosomal inactivation of one of their X chromosomes. You might have heard of something called Bar bodies, for example. The basic idea is that in women, or in cells that have two X chromosomes, one of the X chromosomes gets inactivated. So the idea is that everybody's cells, no matter if you are XX or XY, everybody's cells really only have one active X chromosome. Because if you are XX, one of your X chromosomes gets turned off. So everybody only has one X chromosome active in their cells, is the basic idea, because of this inactivation of one of the X chromosomes. However, it seems like the X chromosome isn't perfect at inactivation. There are some stretches that inactivate really well, and some stretches that actually kind of just get left normal. And it turns out that all the immune genes are on the parts that don't inactivate. So basically, in a cell that is XX, there are two copies of the TLR7 gene that are making twice as much TLR7 protein as someone who is XY. And that difference in gene dose may be part of this because we're going to see differences in signaling due to differences in protein, because this is actually one place where we see a difference in gene dose, because for some reason a lot of these genes don't inactivate the way the rest of the X chromosome does. So that's one part of it. The other part of it that we have realized, and this will come back maybe today, if not today, then on Monday, is that immune cells are super responsive to a lot of hormones. And you actually already know this, though you don't realize you know it, but we'll get there eventually. But immune cells are super responsive to lots of hormones. Somebody wrote, I was noticing in one of their assignments about how they didn't realize that the immune system and the endocrine system were interlinked and all. And I was like, oh, are they? They work together so much. And so the other thing is that different levels of testosterone, estrogen, progesterone, etc., can actually impact function of different immune cells. And so that also is probably playing a role here in these differences in immune responses. But this area is an area that has really only become of particular interest in maybe the past 10 years, or has at least like increased in interest over the past 10 years. Sabra Klein, who's the author listed here, has done a particular amount of fabulous work on this. And one of the papers that was for the assignment due today also talks about gender-affirming therapy in trans men and giving them testosterone and how that impacts their immune system. So that was sort of one of the options there. That paper is super cool. So as I told you about before, there is sort of this group of different types of conditions that all are related to kind of too much immune system, too much immune response. These include hypersensitivity reactions, autoimmune reactions, and transplant reactions. And those differ based on what's the antigen. Is the antigen a self-antigen? Is the antigen a foreign, harmless antigen like pollen? Is the antigen someone else's kidney? In every case, the problem is too much inappropriate immune responses. When we think about dividing up all of the different disease states that may happen with these too much immune system situations, we see the same mechanisms over and over again. And so we can basically classify these diseases into these 4 different mechanisms called type 1, type 2, type 3, and type 4. You can see that there are autoimmune diseases that are examples of type 2, type 3, and type 4 responses. You can also see that when we get to transplantation, the mechanisms we're going to see are similar to type 2, type 3, and type 4 responses. And when we think about allergy, which I really think of as hypersensitivity, we have these 4 types of responses as well. So for a lot of what we're going to be doing today and on Monday is talking about the 4 types of allergy, the 4 types of hypersensitivity responses, kind of what the mechanism is, what that looks like, how that results in a disease, some diseases that are examples of it. But realize that as you start to understand those 4 mechanisms, the mechanisms that you're understanding are going to be translatable into other situations. You know, as you learn how a type 2 allergy works, you're going to actually be seeing the same mechanism that's working in some other types of diseases. But we're going to do them all with allergy. What you can also notice in this image is that allergy, when we think about allergies, there is this sort of unique feature in that this type 1 response is seen in allergy when we are looking at responses to foreign harmless stuff. And we don't really have a type 1 equivalent in autoimmunity and in transplantation. I will also tell you that if I asked you to think of an allergy right now and imagine an allergy, most likely the allergy you would imagine is a type 1 response. Type 1 are sort of the most famous allergies, and they are unique compared to some of the other responses. So the majority of what I'm going to be talking about today is what's up with a type 1 response and how does a type 1 response work. My guess, trying to project out where I'm going to get in these slides, is that we'll do the other types on Monday, but we'll see how time works. And so again, you can also see in the image at the bottom, this is the image from your textbook showing these 4 different types of reactions and how they work. So this is all coming from chapter 14 of your textbook. I'm going to be zooming in on different parts of this figure at different parts throughout class today and Monday, but this is sort of the overview of what it's trying to show you. I'm also sometimes going to show you this version of this table, which comes from a different immunology textbook, where we can see the 4 types of hypersensitivity responses as well, but this one actually has some useful words. The other one just is a picture. And so what you can see is that one big difference between our 4 types of hypersensitivity responses is what the mediator is. And so really what I would say is a key thing to think about, and again, we're going to zoom into this more, is that with types 1, 2, and 3, antibodies are really causing the problems. And with type 4, T cells are causing the problems. And you will also see as we talk through this in detail, the particular type of antibody varies between type 1, 2, and 3. So it's not all antibodies for all of them. There are specific antibody situations for 1, 2, and 3, while number 4 is T cells. And so you can also see the different mechanisms described here. And you can also see sort of the typical manifestations, so situations where these things happen, diseases that involve these particular types of hypersensitivity reactions. And so we'll be coming back to this sort of a few times as well. As I'm looking at what the next slide is, it looks like I've taken the type 1 words from this slide and the type 1 picture from the other one and put them together. So if we start out with type 1 hypersensitivity, we're just going to go in order through our types. One thing to know about the type 1 hypersensitivity response is that the type 1 hypersensitivity response also has another name. And thinking about this other name is one thing that's pretty helpful in thinking about a type 1 hypersensitivity response. So the type 1 hypersensitivity response is also known as an immediate type hypersensitivity response. What that means is that when you are having this response, and you will note that I'm saying this with an asterisk on it, and we'll get to that asterisk in a few slides. When you are having this response, it's usually happening in sort of minutes, often, after you see the antigen. It can happen in up to a relatively small number of hours, but oftentimes we're seeing this in minutes. So one of the things that and that is much faster than all of the other 3 types. And so that immediacy is one of the things that sort of makes this stand out. So if you were, say, looking at an old exam where I had given you some little story and asked you what type of hypersensitivity response is this, one of the things that might distinguish it is if the time I gave you was in minutes, it was probably an immediate type or a type 1. So that's one of the really key distinguishing factors here. The problem in a type 1 hypersensitivity response is an antibody, as we already said, and specifically this is an antibody that is an IgE antibody. So you have IgE causing the problems in a type 1 hypersensitivity reaction. That IgE, if you recall, spends its time attached to Fc receptors, the Fc epsilon receptor goes with IgE. So you can see here Fc receptors on a cell that have IgE bound to it. So remember, we don't have very much free IgE in the blood. It's all stuck to these receptors. And those receptors are primarily found on mast cells. So IgE is basically all stuck to your mast cells. So in fact, one of the other problems in type 1 hypersensitivity responses is the mast cell. So you could, if you told me that the mediator of this was IgE, I would be like, yes. If you told me the mediator of this was mast cells, I'd be like, yes. Either of those sort of work as what the big mediator is, because the IgE is coating a mast cell. And if you remember, we've talked about mast cells having these granules of toxic substances in them. Those granules are pre-made. Basically, the mast cell is just sitting there, doesn't have to transcribe, doesn't have to translate, doesn't have to do anything, has the toxic stuff already in the granule. And so if antigen binds to the IgE, then, which you can see here happening at the bottom, then the mast cell is going to just release its granules immediately. And this, again, can happen in minutes because we don't need transcription, translation. We don't need anything else. Everything's pre-made. The IgE is pre-made. It's already on the Fc receptor. The granules are already made. As soon as the antigen's around, immediate triggering of that mast cell and immediate release of these mediators. And so then we're going to see the symptoms coming from the release of those mediators. And I will talk about what those mediators are in a future slide. But the one other thing I kind of have to mention here briefly before I tell you about those mediators is that, as it says on the title, this is an immediate type hypersensitivity response. I told you one of the really important things about this is the immediacy of this response. And I also told you that there's a little asterisk in my head about that immediacy. And so this is a key thing to also be aware of. Because it is an immediate type hypersensitivity response. Except, as you see here, the key mediator is IgE. How do we get IgE? Where does IgE come from? Yeah, Emily. Okay, we need a B cell to make that IgE, sure. Anything else you know about that B cell if it's making IgE? Yeah, Abigail. Yeah, that B cell had to already have been activated and already do class switching. So somehow, because there's IgE around, we know there was a previous B cell activation, there was a previous class switching. So there's previous stuff that has to happen before this immediate thing. And so that's why there's sort of this asterisk on immediate. Because in fact, really what happens is that this response, a type 1 hypersensitivity response, and you'll see this on the next slide, is immediate the second time you see an antigen. And it's immediate the third time you see an antigen. But it's not immediate the first time you see an antigen. The first time you see an antigen, basically nothing happens, at least symptom-wise. You don't think anything happened to you, but you're kind of having like a setup phase. And then after that setup phase has happened, every other time you see the antigen for the rest of your life, it's immediate. And so you can see this here. So here is this person who the first time they see they're seeing pollen, and it's the first time they see this pollen. The first time they see that pollen, the pollen antigen, is picked up by some antigen-presenting cell. We're turning on a T cell. The B cell turns on the T cell turns on a B cell. The B cell makes IgE. So like all of this happens. IgE starts getting made. It starts getting bound to the mast cell. All this is happening. This person doesn't know this is happening unless they've taken immunology and spent way too much time thinking about this like me. So all this is happening the first time the person sees the antigen. They don't really know anything's going on. But by the end, we have these mast cells that are pre-coated with IgE. Now, the second time the person sees that same antigen, those mast cells immediately degranulate. Those mast cells that are coated with IgE immediately get triggered and immediately release their granules. And so when we think about the immediate type hypersensitivity phase, we sometimes will say that it's only immediate in like the effector phase, in the second phase. There is also this earlier phase called the sensitization phase, where you become sensitive to this process. It's only later that, in fact, you start to have this response. And so the place where people are most familiar with this is with bee stings and bee sting allergy. In general, the first time you're stung by a bee, nothing happens. If you're going to have a really severe bee sting allergy, it's going to be the second time you're stung by a bee. And so there's sort of this, question of if, like, do you have a bee sting allergy? And there's sort of this question of like, I don't know, I've never been stung twice. Maybe I've only been stung once. Maybe I'm just ticking time bomb. I don't know. So that's the one where people have sort of heard of this before. The other thing we need to sort of think about here is what exactly these products are in the mast cell that are leading to all of these symptoms, that are leading to all these physiologic effects. And the mast cell has a bunch of different products in the granule. So there are tons of products inside the mast cell granules. Many of them are enzymes. You can see some of those enzymes listed here, like tryptase, chymase, cathepsin G, whatever. The point of those enzymes is to try to, break down a parasite, break stuff down. So we're trying to degrade whatever biological molecules. This also can be really useful if we need to degrade a wound or something. So the mast cell is going to release a bunch of these enzymes. The mast cell is also going to release a bunch of different lipids or other small molecules, like you can see here, that are directly able to kill parasites. The most famous of these small molecules that we see is this one, which is histamine, everyone's unfavorite molecule. So one of the big things in those granules are histamines. Some of those enzymes are there as well. We've also got cytokines and chemokines that are in there, and there's a wide variety of cytokines and chemokines. And finally, we have some of these small molecule lipids. So we have what's listed here as the lipid mediators. In fact, you can see here is here's one kind of lipid mediator, which is called arachidonic acid. It can actually be modified through biosynthesis pathways to things like prostaglandin D and leukotrienes. Here's another place where we have lots of interaction with endocrine things because some of these have important endocrine effects. Leukotrienes are often thought of as particularly problematic things with mast cells, You can also see that this shows, but both leukotrienes and prostaglandins can be involved in inflammation. You can notice that this little image, and this is actually the whole reason I have this image here, shows the enzymes that are involved in this conversion. So you can see this one says 5-lipoxygenase, and this one says cyclooxygenase as the enzymes. Cyclooxygenase can also be abbreviated COX, C-O-X. Specifically, this is actually called COX-2. And COX-2 is the target of aspirin. So salicylic acid, when you're taking aspirin, you are actually blocking this pathway right here to block inflammation. So I like to point this out as a sort of a tie-in to things that you've thought about previously in your life. So these are, like I said, some of the mast cell products. So you can notice that the mast cell is making a lot of products that are involved here. It's not just one thing. You can also see this happening here. So here's my mast cell, and it's making a bunch of these different types of compounds. These small molecules like histamine, these lipids, cytokines, enzymes. You can see there are lipids again. And we can talk about what exactly each of those things are responsible for in this process. Before I do that, I want to point out, I get really into talking about some aspects of allergy because I am allergic to literally everything. In 2025, I've been actually doing a lot better than I had previously because I started a new regimen of allergy medicines. So I take 3 allergy medicines a day. One of the allergy medicines I take every day is Flonase. And when Flonase really came out, their commercial was really funny. And I want to tell you what their commercial said, and then we can look at this slide. Because what their commercial said was, Flonase is really great because it blocks 6 things involved in allergy instead of 1, and 6 is greater than 1. And if you even still look at a Flonase package, it says 6 is greater than 1 as like one of the big things on the package. The point there is that most previous allergy medicines just block the histamine, whereas the Flonase is actually blocking all of the things. That's where they're getting their 6 is greater than 1 business from, is they're saying multiple of these things are getting blocked instead of just 1. And so this is sort of very much something that is being thought about in the pharma industry, or sort of all of these different products that are coming from mast cells. And we figured out ways to block one of them, histamine, pretty early. And sometimes we just block that one, but we let all the other ones go. Sometimes now we're actually able to do a little bit more broader blockade. And so many of the allergy medicines people take are antihistamines. They're just blocking the histamine, but letting all the rest of this stuff happen. And you can see some of the results of these mediators and what these different mediators do here. So you can see that one of the things that happens is we see changes in the blood vessel, we see changes in the vasculature to give us vasodilation. You've seen vasodilation earlier this semester. So where have you seen vasodilation, and why is vasodilation useful in immunology? Yeah, Emily?
Like local inflammation, and so that way the immune cells can come to the site that needs help.
Yeah, so we're going to be able to get more immune cells to that location in its sort of classic inflammatory response. There are a lot of ways that this is going to look like a classic inflammatory response. And if you think about what it feels like to have an allergic reaction, it feels like sort of classic inflammatory responses in a lot of ways. We also have changes to the vasculature to make the vessels more leaky. So we have vasodilation and increased permeability. Here it's called vascular leak, but it's the same increased permeability you saw before. Basically, the blood vessel gets leaky. So cells, proteins, et cetera, can get out and go into the tissue. So you can also see down here, we've got inflammation happening. That is a big piece of this. You can see down here with the enzymes, we also have some tissue damage. And that tissue damage is largely because either we're trying to damage sort of parasite, this is because this may have evolved to deal with parasites, or it's sort of involved in kind of the healing of the wound and trying to make sure like you don't have a clot in the wrong place or something like that is why we have this tissue damage. We also see one other pretty key thing in a lot of cases as a result of these mediators. And if I were to take that and put it in a super big picture way, I would say that it is smooth muscle contraction. And so many of these mediators lead to smooth muscle contraction. There are different kinds of smooth muscle in different parts of the body. And so smooth muscle contraction can end up being different things in different parts of the body. So sometimes that smooth muscle contraction can lead to constriction of the airways or bronchoconstriction because we're basically contracting the muscles in the respiratory tract. Sometimes that smooth muscle contraction can be in the GI tract, and we're seeing those smooth muscles contract and we're seeing GI symptoms. We also very often, in a lot of these cases, sort of see a response that, and this is related to that smooth muscle contraction, that seems to be kind of a get rid of contents of body. Like, I have a bad thing inside me, let's expel whatever we can expel. If that is in something that's, thinking in the respiratory tract and you're trying to expel, you're going to have a lot of mucus, you're going to be sort of wheezing and breathing fast, and you're really going to be trying to expel stuff in the respiratory tract. If it's in the GI tract, you're going to have stomach cramps and vomiting and diarrhea and all of that kind of stuff. And again, really they're sort of like a expel this parasite. In whatever situation. So you sort of see what these products of mast cell activation are. The other thing to be aware of is that when we think about an immediate type hypersensitivity response, this is a big part of what we think about in terms of these immediate effects. But in particular, the cytokines also will have an effect longer term. So they will keep doing stuff after that immediate response. And so a lot of what people think about when they think about a type 1 hypersensitivity response is sort of this early phase response. And if you have, some cytokines made that one time in response to some kind of allergen, whatever, that's fine. But those cytokines will keep acting on other aspects of your immune system and may influence other aspects of physiology. And so oftentimes there are longer term late phase responses to an allergen as well. And so we do often see kind of later phase immunologic and physiologic responses to allergens that we don't often think as much about. And honestly, they're not like a giant deal if you're getting that allergic reaction one time. But if you are seeing that allergen all the time, those late phase sort of structural changes to your immune system can be a real problem. And you'll see an example of that in a later slide. And so you can also kind of see this here. So we can look at, this looks terrible projecting. It looks fine on my screen. So we can see that immediately after this person saw this antigen, they get this little welt. But in fact, if you actually look long term, there is sort of general inflammation and swelling outside of that 15-minute time period. And so this is more of a late phase response that's happening due to the cytokines as opposed to some of the other more quickly acting mast cell products. As I have mentioned a number of times, there seems to be a pretty big genetic component to this. And so there are some individuals who we know that are what are known as atopic individuals, or these individuals have atopy, which seems to be a predisposition towards making a Th2 response. When your body is making an immune response, for whatever reason, you are more prone to make Th2 than anything else. That might be because of some change in a cytokine receptor, so you make Th2 cytokines easier, or it could be all sorts of things. We don't exactly understand. This definitely has some genetic component. It may also have environmental components and be related to the hygiene hypothesis that we talked about last time. But in the end, there are some people who are just strongly predisposed to a Th1 response who see these type 1 responses very often. They tend to end up with things like asthma, allergy, dermatitis, etc. There's also there are certain physical traits that we tend to see more frequently in atopic individuals. If you would like to imagine kind of a sort of classic atopy example, here you go. Because I am super atopic. And so part of this is likely very much a genetic predisposition. When we think about the genes that are part of this genetic predisposition, we see things like MHC, T cell receptor, cytokines, FC receptors, things involved in making those lipid mediators, etc. So it's sort of all stuff that really makes a lot of sense. It's, things involved in a lot of these situations. one thing that is sort of, that will sometimes be unique is that as we're going to see starting on the next slide, there are kind of some different directions a type 1 hypersensitivity response can end up happening. So, in some people it's a skin allergy, in some people it's a respiratory allergy. And that might be because you have the atopy genes and a weird skin gene, while somebody else who has the atopy genes and a weird lung gene gets lung allergies or something like that. So often it's sort of a combination, and there's probably some, so here, this one is showing, for example, specific stuff for asthma on the right, and it points out that, well, one of the genes involved in asthma is involved in airway physiology, but the rest of them are involved in like hyperactive immune responses. So it does sort of end up working a little different in different individuals. Yep, Emily.
So, I apologize if I'm getting ahead of it, but we make antibodies to anything that's foreign, right?
Mm-hmm.
So, if like I'm allergic to peanuts, then I would already have the antibody to peanuts. Is it because it was a danger signal the first time?
So some of this we talked about last time.
Okay.
But a lot of it has to do with what happened in your first encounter,
Okay.
and how that might have been a first encounter that was a little different than normal. But it also might be maybe you're just more likely to make inflammatory cytokines.
Okay.
So maybe your trigger for making an inflammatory cytokine, maybe your PRRs don't take as much as everyone else's. And so there would, and for you, be a danger signal, but that wouldn't be enough for anybody else.
Okay, but then also like there's
Mm-hmm.
like, probably this much of the antigen, but then it like is it that when you already produce cytokines, then like those cytokines activate other things, and it's just like
So part of it is,
explosion?
yes, so part of it is definitely that those, small, one cytokine is going to, there are downstream effects. It is certainly a cascade type of thing. The other thing that you want to think about is, the first time that peanut might be turning on like one cell, but that cell's then going to have a clone army later on. And so now you're going to have a lot more cells involved. But I would definitely say take a look at some of the stuff from last time as well, because that's going to help a lot of that. All right, so the one thing we haven't really talked about with type 1 hypersensitivity here is what's the antigen that's involved. And so this slide has some examples of type 1 hypersensitivity antigens. So you can see that type 1 hypersensitivity antigens can include ryegrass, ragweed, timothy grass, birch, a variety of drugs, peanuts, seafood, eggs, milk, bee sting, wasp sting, ant sting, cockroaches, dusts. This is cat hair, this is dog hair, any of your animal allergies. This is a latex allergy, this is a mold allergy. So you can see there's a pretty big range of antigens that are associated with type 1 hypersensitivity responses. As you're looking at this, you see penicillin listed. There are actually multiple kinds of penicillin allergies. There is actually a penicillin allergy type 1, type 2, type 3, and type 4. So penicillin can be any of the 4. I tell you that because I'm going to give you penicillin as one of the examples and one of the other ones. So like, don't get too hung up on seeing penicillin here because it's going to show up in other places as well. And you can see your textbook, instead of putting this in table form, puts all of this in picture form. Note that it mentions sort of cockroaches, but also it mentions dust mites. I think sometimes when people have a dust allergy, they don't realize you actually are allergic to something called a dust mite. You're not allergic to the dust itself, which is a little bitty microscopic invertebrate that eats the dust. So it has certain antigens as part of it. And so in fact, there is some similarity with other with cockroach allergy or something like that, because they are all little invertebrates. I could freak you out a lot by saying various things about dust mites and things about mites. I won't right now. I won't, for example, tell you how many mites currently live on your eyelashes. But if you really want, but you can Google it. You can Google it if you would like. So the point of this slide, other than being that I get to freak you out about mites, is that if you think about if you imagine allergies to a lot of these different things, you don't necessarily, you imagine things that are different. You're sort of, sometimes people look at me and like, I'm sorry, you're telling me that a peanut allergy and what happens to me in response to ragweed are the same thing? Those do not look like the same thing. Like these seem very different. And that doesn't seem like, those don't necessarily seem similar to what's going on with cat hair. Like cat hair, peanut does not strike a lot of people as the same thing. And here I'm telling you, they're all the same immune response. And so we kind of have to think about that. And the reason why we think of sort of the thing that's key with all of this is that they are all the same response. They are all exactly the same immune response, but that immune response is happening in a different anatomic location. And it turns out the result of the same products of a mast cell, the result of that mast cell doing the exact same thing in different parts of your body, give you slightly different outcomes. So it's always a mast cell misbehaving, but because that mast cell is in a different location, and the reason why, of course, that mast cell is misbehaving in that location is because that's where the mast cell lives. And that's where you saw the antigen. So about some of this is also that you see some of these antigens in different anatomic locations. Will lead to different types of symptoms. And so you can see, we've got sort of anaphylactic shock. That's type 1 hypersensitivity. We have a lot of skin issues. We have asthma. We have food allergies. We have hay fever. All the same exact response. They're just mast cells doing their thing in different anatomic locations. So if we see this response in the skin, you can see that the mast cell can be in the skin. It can influence the blood vessels just in that location in the skin, not blood vessels throughout the whole body. And so in that little location of the skin, you're going to get changes in vascular permeability and swelling, and you're going to get like a little swollen mark. You're going to get like this little thing on your skin, a little bump, that's officially known as the wheal and flare response. And so basically you're seeing mast cells that are responding and giving you that little bump. Also just fun facts, because who doesn't love fun facts? When you get that little bump with a mosquito bite, it's actually because you are making a type 1 hypersensitivity response to mosquito saliva. Any of those sort of like those little those little bumps that you get in different situations, it's just the mast cell acting on the vessel right there and leading to liquid and cells coming out of the vessel right there and giving you that little bump that you see on the skin. If this exact same thing happens in the respiratory tract, here's the mast cell, it's acting on the blood vessel, and we're going to see that same kind of influx of cells. But now we're going to have a lot more kind of epithelial and smooth muscle that's in that area, which we didn't have as much, that we weren't seeing as much in the sort of right there in the skin. And so we're going to have, decreased, we're going to have changes in the blood vessels, but we're also going to decrease the diameter of our respiratory, of our airways. And we're going to start expelling those airway contents. And so we're generally going to see sort of constriction of the throat, constriction of the airways, difficulty swallowing and breathing, and wheezing all coming together. We're going to be making a lot of mucus because we're trying to get the thing out. And so we're trying to make a lot of products to get out so we can sort of, have a little waterfall of mucus of pushing whatever it is out. All of this is coming, again, directly from those mast cell products. It's just the mast cells are doing this in a different location. If this happens once, that's, not great. If this happens many times, you can see that we now start to get larger scale changes to that airway. And so this is an airway in an individual where we don't see asthma. And so you can see we're supposed to have this sort of thin layer of this smooth muscle. You can see some of the tissue under it. And you can also see this part. And what's important about this part? Right here, this right here. That's the empty part where the air goes, right? That's where the breathing happens, right? As time goes on, that smooth muscle layer gets thicker and less able to, less flexible. You can see there's a bunch of other changes in the structure because of those cytokines that are being made for a long period of time. But you can also see this mucus production and all this mucus. And what do you notice in this sort of person who's been making this response for a long period of time that's in asthma? What are you noticing about this area?
Not enough empty space.
Yeah, it's all mucus. There's no place for the air to go. There's no place for the breathing to go. And so you can kind of see that this is a case where those cytokines in their long-term response are going to be causing a lot of other issues. And so basically what you should have seen here is this difference in what happens if the mast cells are in the skin or if the mast cells are in the airway. And so is the first place you see the antigen in your skin? That might be like your mosquito bite. Or is the first place you see the antigen in your airway because you breathed it in, like pollen or something like that? We can also think about food allergies. So here we're thinking about mast cells in the GI tract because the first place you're seeing the antigen is in the GI tract after ingesting it. And so now you can see the mast cell in the GI tract. We're going to influence blood vessels in the GI tract, but we're also going to influence sort of GI tract cells and GI tract smooth muscle here. And so now we're going to start to see things like vomiting and diarrhea and sort of stomach cramps, all of those types of things because of all those effects on the smooth muscle. One other thing to note is that sometimes when you start to see antigen in the GI tract, that antigen can then get a bit into the bloodstream. And so you might see some effects elsewhere. In particular, this says urticaria, or urticaria. That's the fancy word for hives. And so there you've actually had that antigen start going throughout the blood and you're starting to see some inflammation in different levels of the skin. One other thing to note about food allergy, there are lots of questions. There's some questions about this and a future slide that people always ask, and my answer is I am not an MD, I don't have the answer. And so there are some food allergy questions that people can ask that I can like make up an answer for, but I don't actually know. And one thing that I will also say is that sometimes with food allergy, the symptoms get a little weird, partially because of that antigen in the going into the blood, but also because it's really hard when you're eating something to not also have it hit the respiratory tract. So if you imagine eating something, sometimes you're going to breathe in a little bit of that antigen as well. Depends on how much digestion the antigen takes before causing the problems. And so sometimes like you'll see a situation where you're getting kind of multiple anatomic locations getting hit because of how you have experienced that antigen. And so here you can see some of the sort of many different aspects of different types of allergic reaction. And in fact, this also points out, sometimes food allergies end up being a rash, and that's usually because of the touching the food. Sometimes they end up being kind of more wheezing, and that's actually because it's really the problem has been the amount of antigen you inhaled when you ate that food. And so sometimes you can see some different manifestations of food allergy based on the fact that we don't often just interact with that antigen in one place. You might see the symptom in the mouth versus in the intestine or something like that. So food allergy can look like a lot of different things. The other thing that is on this slide that many years I get questions about is exercise-induced allergies or exercise-induced wheezing anaphylaxis that is listed here as being related to food allergy. I got nothing. A publisher sent me a copy of a book called Exercise Immunology yesterday that I could potentially read. It's on my computer now, but I haven't read it yet. So maybe I'll know for next year, but I got nothing now. So if you have questions about what happens with exercise-induced asthma and things like that, I cannot help you. And so again, you can kind of see these details with food allergy. You can also think about our celiac example from last time, where normally in the GI tract, we often will have this situation where sorry, let me go back. Normally, if you eat an antigen, the first time you see that antigen is by GI tract, you often get a regulatory T cell response. As we talked about before, I have some information about that here as well, that oral tolerance, because you don't want to make immune responses to food. So if the first time you see an antigen is by ingestion in a normal, healthy situation, yes, food allergy, there obviously is a problem here. But if you see it the first time by ingestion, oftentimes you won't have a response. But if for whatever reason you see that allergen in the skin for the first time, you might say make some other kind of T cell response, like a Th2 response. Now you have Th2s, and you happen to then see that same antigen again by ingestion in the GI tract. Now you're not going to make Tregs anymore because you already made Th2s, and now you're going to potentially start to have a food allergy response. And so I told you guys about the issue with peanuts and increasing number of peanut allergies, that one of the things that we think it may be associated with it is increasing number of lotions that have peanut antigen in them. And so more babies are seeing peanut for the first time on their skin instead of through ingestion. And so not using lotions with peanut antigen, and also feeding kids peanuts as early as possible to sort of make sure that they're getting a tolerogenic response does seem to be really key. So one other thing I want to mention in terms of anatomic locations and type 1 hypersensitivity is what happens if the antigen is in the blood. And so if we end up not with the antigen in the respiratory tract or the GI tract or the skin, what do we see? And if we end up with that antigen in the blood, then that's where we have anaphylactic shock. And with anaphylactic shock, we are now seeing mast cells throughout the body, throughout the blood, getting activated. So you got mast cells getting activated everywhere. So they start to impact the GI tract with contraction of smooth muscle, stomach cramps, all of that stuff. They also hit the respiratory tract. They also hit the vasculature generally throughout the body. And so we see all of this constriction of throat and airways, difficulty swallowing, breathing, wheezing, stomach cramps, vomiting, diarrhea, as well as swelling and loss of blood pressure and irregular heartbeat and all of those things. This is a very severe response. You can see all of the different sort of anatomic responses that are happening with anaphylaxis. This is sort of that response that people are trying to prevent with their EpiPen, because they're trying to sort of make this not happen, because this is immediate type with all of these mast cells acting, and really leads to pretty severe outcomes in individuals. And so if that antigen is in the blood, we're going to see many different anatomic locations responding. So two other things that I want to kind of point out that are interesting. One of them, of course, is when I start to get really annoyed and think, why the heck is my immune system think pollen is bad? Why is it so mad about pollen? Pollen never did anything to me. Why are we making an immune response against pollen? And it seems like if we look at many of the different things that are antigens in the case of allergy, they are relatively cross-reactive with some antigen in some kind of parasite. And so it seems as though these are antigens where we might be sort of predisposed to making a Th2. We just never see them on the helminth, so instead we're seeing them on these other sort of similar cross-reactive types of responses. There is and so sort of this cross-reactive allergen and helminth is one big idea here. There is a sort of competing hypothesis that also has some interesting ideas that There also is a fair amount of cross-reactivity between allergens and things like snake venom and insect venom, which are toxins that you have to get rid of right away. Your body needs to, like if you get bit by a snake and there's snake venom, you need to expel it immediately or else you're going to die. And in some ways it's like, wait, but this immune response seems to be all about expelling things fast. And there also seems to be some cross-reactivity with those venoms. Remember the difference between venom and poison, that they are not the same thing. Venom is when something bites you and you die. Poison is when you bite something and it dies. Is it something that you ingested, or is it something that someone else bit into you as a toxin? So specifically, these are snake venoms that are part of this. But the other thing that I wanted to kind of tell you a little bit about is as sort of a reminder that when we are talking about these responses, we are talking a lot about responses that are specific to different anatomic locations, right? So I've told you about how the issue with the respiratory allergies is that you have mast cells in your respiratory tract that are coated with IgE, that when you breathe in the antigen are going to lead to IgE and things like that. And so in talking about all of this and thinking about all of this, I have kind of a little interesting case story to tell you that I think is sort of instructive for all of us to think about. So as I have told you, I'm allergic to everything. I have a lot of allergies. And 2 years ago, a year ago, might have been last fall, I don't remember when it was, I finally went to an allergist. I'd never actually gone to an allergist to have certain aspects of my allergies diagnosed. I finally went to this allergist and I was so excited. And he found out I was an immunologist, so he tried to talk immunology to me. And he went on and on about Th33 cells. And I'm like, yeah, those don't exist. But also, he did a classic allergy test on me. He wanted to test to see, we were going to see which pollen is it that's causing me all the problems. And so he did this allergy test where basically what they do, it's called a patch test, is they inject you with small amounts of various antigens in your arm, and you look for that classic wheel and flare response that I showed you. And I suffer with allergies. It is very clear. So, and in fact, the wheel and flare, when they inject into your skin, you get it within a few minutes. It is definitely an immediate type hypersensitivity response. So that is my arm. Again, hard to see with this thing. This right here, where is it? That right there is a positive control of histamine, where they just put histamine straight into my arm. And you can actually see this little welt there. All of these other things are other types of pollen that they injected me with. And my response was 100% negative. I definitely can make a response because that histamine, I respond to that histamine. And I wanted to kind of rip my skin off. It hurt so bad. And I was just like, ah. But the rest of them, absolutely nothing. And so you can see all of my absolutely nothings here. And we were like, this is weird. But he was like, well, that happens sometimes. It's okay. He's like, I see this sometimes. So we're going to do a next step. And so his next step was that he did he drew some blood and he sent it to a lab to have them look at my IgE levels in the blood that responded to different allergens. And this is a screenshot from my health website of the data from my respiratory, from how much IgE I have in my blood against all these different things. So you can see all the different types of pollen they tested me against. And you can see, it was impressively zero. It wasn't like there was a little bit of background. It was zero across the board. And so we looked at this and he's like, yeah, we see that sometimes. He's like, so my prescription to you is to start taking 3 over-the-counter allergy medicines every day. Because I can't diagnose you. I got nothing. These are literally the data. And again, he was like, that happens sometimes. And in some way, and like, this is the classic, some of you guys are shaking your head like, yeah, I've had this allergy test. Like, this is the classic way of one doing allergy testing. And I want us to think about it for a second and why both he and I were I was annoyed because I wanted to like get my allergies gone. But he and I were both like, yeah, that happens sometimes. Neither of us were actually completely shocked by this. And so I want to think for a second about why neither of us were completely shocked about this. When I have problems with all the trees, what anatomic location do I have my tree problems with? Yeah, Miles. Mostly my respiratory tract. I get super stuffy, I cough, like I have some runny eyes. It's all like right here, right? What mast cells did he test? Right here? He tested the mast cells in my arm to see if they were sensitive. Well, I don't have a problem in my arm. What mast cells, what did he test here? And what specifically was he testing there? What is it? What was it? I see you kind of saying it. Go for it. Not so much the digestion. What's he testing here? IgE. What did we say way back at the beginning of the semester about IgE levels in the blood? There really aren't. IgE doesn't spend time in the blood. IgE spends all its time bound to mast cells. So guess what? I had no free IgE in my blood. Shocker. It was bound to the mast cells, potentially the mast cells in my nose. And so one of the things that I think is really, that I started thinking about much more that was really interesting is sort of the fact that we can't test every anatomic location for allergy. And so I didn't think I realized how like weird that was about allergy testing until this experience. One of the conversations that we had, and so again, maybe there's a different immune response that's circulating versus in the nose or things like that. And then so, in fact, what he and I both talked about in one of our conversations was he's like, oh yeah, he's like, probably the reason why you're testing like totally negative for everything is because you just have all the IgE and mast cells in your nose and I didn't test that. He's like, so there are some experimental situations where we actually put people in like a hospital room and we blow pollen up their nose to look for allergic reactions. And he's like, so in experimental settings, we can do that and prove that people do have allergies, but I don't think it's worth it to do that. I just think you should take over-the-counter allergy medicine. And again, this did not feel particularly nice as an answer, and it just made me very annoyed. But at the same time, it made perfect sense with what we currently know about immunology. And it's sort of an interesting thing to think about when you're thinking about some of these clinical manifestations and clinical testing, and do some of the tests like a patch test on the arm. I mean, yeah, sometimes they're definitely positive. But if they're negative, what does that actually mean? And how is that important for us in thinking about sort of diagnosis in the clinic? Yeah, Julia, so I had a quick hand.
My question was just like, that makes perfect sense, but then like, how are some people very like they just happen to have a lot more free?
And so, and then they must have more free IgE not bound,
somehow.
Or their mast cells throughout their body must have been coated, whereas mine was really specific. And why that is, who knows? But like as I started thinking about this when it happened, I was like, oh, this actually like makes perfect sense with what I teach. In terms of the importance of subcellular location. And that's like the area where a lot of immunology is going in terms of location-based immune responses. Yeah.
So if you do respond to these, to IgG and to these tests in the blood, does that mean that your mast cells are, because there's more IgG in the blood, it's more likely for that
I don't think that they've done enough studies to characterize the two groups of individuals to answer that question, because you'd have to actually prove that, you'd have to do the pollen experiment on me, right? Prove that, show that allergy, and then test me versus somebody who's positive for our anaphylactic shock response. And those are not easy experiments to do. Yeah, no problem. All right, so remember your assignment by 5 if you haven't given it to me already. We'll do the type 2, type 3, and type 4 on Monday. So I will see you guys then.